Hello my dear students, welcome back to Biology at Priyajit. Imagine observing a tissue under a microscope hours after removal. What happens? The cells start self-digestion, bacteria cause decomposition and the tissue structure gets destroyed. So how do scientists preserve tissues in a lifelike state? That's where fixation, embedding and microtomy come in, the foundation of histology and histopathology. In this class, we will learn fixatives, fixation procedures, embedding, microtomy like cryostat, artifacts and exam important concepts useful for undergraduate and postgraduate, viva and other theoretical competitive examinations. Let's begin. Why tissue preparation is necessary? Fresh tissues cannot survive outside the body, so they undergo autolysis. Auto means cells. Cells contain enzymes that start digesting their own structures after death. Example, think of fruits becoming soft and decomposing over time. Putrefaction, microorganisms like bacteria and fungi cause tissue decomposition. Therefore, tissues must be preserved immediately. The important steps in histological preparation are fixation, dehydration, clearing, embedding, sectioning and staining. Remember this sequence: fix, dry, clear, embed, cut and stain. Easy mnemonic is fresh doctors carefully examine cell structure. That's to remember the concept, to memorize the concept. This is the flowchart showing the process of tissue fixation. Tissue processing. What is tissue fixation? Fixation is the process of preserving tissues as close to the living state as possible by preventing autolysis and decomposition. Simply, fixation is equal to preservation of tissue structure. Objectives of fixation: preserve tissue architecture, maintains normal arrangement of cells, prevent autolysis, stops enzymes from digesting cells, prevent putrefaction, kills or inhibit bacteria. Hardening tissue makes tissue firm enough from the section cutting. Preserve chemical components, protects proteins, DNA, RNA, lipids and carbohydrates. Importance: improves staining. Good fixation gives better staining quality. Long-term storage preserves specimens for teaching, research and diagnosis. What is fixation important? Why is fixation important? Preservation plus prevention of autolysis plus hardening plus staining plus storage. This is the importance of fixation. Principles of fixation: fixatives work mainly by protein coagulation. Proteins precipitate and harden. Examples: alcohol or mercury chloride. Cross-linking proteins, proteins become interconnected. Example: formaldehyde. Oxidation changes proteins and lipids. Example: osmium tetroxide. Altering colloidal state changes tissue chemistry. Remember: coagulate, crosslink, oxidize and stabilize. Methods of fixation or fixation methods. Chemical fixation: use chemicals called fixative. There are two methods. Immersion fixation, a tissue placed inside fixative. It requires tissue thickness of 3 to 5 millimeter and fixative volume should be 10 to 20 times of the tissue volume. Advantages: it is simple and economical procedure. Disadvantage: it needs more time, so it requires slow penetration. Perfusion fixation: fixative injected through blood vessel. It is uniform fixation and it's a rapid process. It uses experimental animals and neurohistology. Physical fixation: it uses physical methods like heat fixation, freezing fixation and microwave fixation. Ideal fixatives should prevent autolysis, prevent putrefaction, preserve morphology, penetrate rapidly and cause minimal shrinkage, improve staining and be economical. But remember, no single fixative is perfect. Fixation, the most critical step in tissue processing. Fixation preserves tissue structure. It locks cell components in proper place. Poor fixation gives poor result and it is critical for reliable diagnosis. Chemical fixatives. Chemical fixatives are of two types: simple fixative and compound fixatives. Simple fixatives contain one chemical such as formaldehyde, which acts by cross-linking proteins, used in routine histological procedures. Glutaraldehyde, it is used in electron microscopy. Its advantage: it is responsible for excellent ultrastructure preservation. Ethanol and methanol, it acts by protein coagulation. It uses blood smears and cytology. Acetic acid is best for nuclear fixation. Disadvantage, it causes swelling. Osmium tetroxide, it fixes lipids and used in electron microscopy. Compound fixatives, they contain multiple chemicals like Buin's fluid, which are used for soft tissues, Carnot's fluids for chromosomes, and Jenker's fluids for bone marrow treatment. Chemistry of fixative: protein coagulation, alcohol removes water, proteins denature. Cross-linking, formaldehyde creates a methylene bridge which results stable tissue. Oxidation, osmium tetroxide fixes lipid. Acidic action, acetic acid preserves nuclei. Physical fixation, freezing fixation, rapid freezing preserves enzymes and lipids. It is used in immunohistochemistry, enzyme histochemistry, and frozen section diagnosis. Its problem: ice crystal artifacts. Microwave fixation, it is uses microwave energy. Advantage: it's a rapid procedure and it needs better preservation, and its disadvantage is risk of overheating. Choice of fixatives: it is used in routine histology. Formalin is used. Electron microscopy, glutaraldehyde is used. For lipid, osmium tetroxide is used. For chromosome study, Carnot's fluid is used, and for freezing fixation is done for frozen sections. Fixation artifacts. Artifacts are the artificial changes produced during fixation. Examples include shrinkage caused due to alcohol treatment, swelling caused due to acetic acid treatment, formalin pigment due to brown black deposits of formalin, ice crystal artifacts occurs due to slow freezing. Over fixation, tissue becomes brittle. So prevention: how can you prevent the process? It is the correct tissue size, proper fixative volume, immediate fixation, and buffered fixatives. Embedding means enclosing tissue in a solid medium for support. Objectives: it supports tissue, it maintains tissue orientation, and it enables thick section cutting. Embedding media: paraffin wax, it is used in routine histology. Resin is used in electron microscopy, and OCT is used for frozen sectioning. Microtomy: what is microtomy? Process of cutting thin tissue sections. Instrument required, that is microtome. Rotary microtome, it is rotary in routine process. Sliding for large sections. Cryostat, it is required for frozen sections. Ultra microtome, it is required in electron microscopy. Common problems in microtomy: wrinkled sections, it is responsible due to dull knife. Remedy is serpent blade. Compression, it adjusts knife angle. Chattering, for hard tissue. Ribbon not forming, incorrect wax hardness. And fragmentation, overheat, dehydration. Cryostat is the microtome inside refrigerated chamber, and it needs temperature between -20°C to -30°C. It uses rapid diagnosis, enzyme studies, immunofluorescence. Advantage is fast diagnosis during surgery. So the conclusion, we can say that good fixation plus proper embedding plus accurate microtomy gives excellent histological preparation. That means better diagnosis, better research, and better microscopic interpretation. If this class helped you understand histology in an easier way, like the video, share with your friends, and subscribe for more UG, PG, Zoology, and Histology classes. Thank you.